There will be more than 300 million of individuals with diabetes in 2030. There will be more than 500 million of individuals with obesity. And the 65% of these individuals, these patients, will die because of any cause of cardiometabolic disease. We create our company trying to bring light in these situations, trying to identify which of these patients are in a higher risk of developing a cardiovascular disease. So from these metabolic-based conditions, which are the worst, or which is the worst molecular signature of future cardiovascular disease. So as you know, LDL cholesterol, so known as the bad cholesterol, it's the main key to assess the cardiovascular risk in the future. So it's the biomarker of cardiovascular disease. LDL cholesterol, it's known as the bad cholesterol. In fact, it's a structure, it's a lipoprotein particle. It's a particle that nature has developed to transport lipids through bloodstream. Cholesterol, it's transported in this structure that it's a low density lipoprotein. It's not very dense. And it's the main responsible to transport lipids from the liver to the cells to be used as a component in the structures, or used as a precursor of hormones, or used as energy. So lipoproteins are, in fact, transporters of lipids that any cell of our body needs. And on the other side, there's the high density lipoproteins known as the good cholesterol, that it's the structures that bring back the lipids accumulated in the arteries to the liver to be excreted. So in a healthy artery or in a healthy system, it might be an equilibrium between these lipoprotein species of transporter lipids. But LDL cholesterol, it's indeed a causal of cardiovascular disease. LDL cholesterol can be accumulated in the arteries, accelerating this pathology that it's known as arteriosclerosis. And this pathology, it's subjacent to any cardiovascular accident in the sense that this atheroma plaque can be can break and be associated with a future stroke, or can be closing the vessel and produce a ischemia, etc. So these atherosclerotic lesions are responsible of cardiovascular disease in the future. So the concentration of LDL cholesterol is indeed a very important parameter to know if an individual is at higher or not risk. But if we take a look on the cholesterol levels of those individuals that have already had a cardiovascular accident, at least in the 50% of the cases, the cholesterol levels are correct. So it's true that when there's elevated cholesterol levels, then the individuals are at higher risk, but it's also true that there's an hidden risk that it's not known, and it's considerably high, that it's not associated with the LDL cholesterol level. So in this case, as a biomarker, LDL cholesterol, it's not good enough. And we create our company trying to bring technology to these patients that are at higher risk, but their levels of LDL cholesterol are correct. So we applied our technology. We came from a metabolomic platform in a clinical environment, trying to apply this technology, metabolomics, based on nuclear magnetic resonance specifically, to reveal what's happening beyond LDL cholesterol. So, as you know, metabolomics is, from all the omics sciences, the closer to the phenotype. Metabolomics is the study of the conditions of biological fluid through the molecules that are present or are constituting this fluid. In genomics and proteomics, these are what is potentially happening to an individual, that it's contained in their genes, but in metabolomics, it's actually what is happening, because it's the real product between interaction on genomics and environmental factors. So we came from the study of pathology or conditions through metabolomics, through the study of molecules that are constituting any biological fluid. So there's more than one approach that can be used to study biological matrices through metabolomics, and one of them is the nuclear magnetic resonance. Nuclear magnetic resonance, that it's a magnet mainly, can be high throughput, so you can profile a lot of samples per day and with a lot of information per sample. It's competitive in terms of prices. It's quantitative, it's robust, it's scalable. You can use a spectrometer here and spectrometer in Chile, and you will obtain the same results. It's automated, it's fast, and it can be used in both modes, as a discovery approach or a targeted approach simultaneously. So that offers a very promising technology for monitoring, for bioscreening in clinical trials or in clinical practices. So in terms of which kind of metabolites you can access or you can measure by using nuclear magnetic resonance, it's also interesting because you can have information from small molecules as amino acids, small sugars related to metabolism function or energetic metabolism, etc., but also macromolecular complexes, such in the case of lipoproteins or even glycoproteins that are inflammatory markers associated with future cardiometabolic disease or cancer disease, etc. So it's a kind of a global bioscreening tool that can give you information from small molecules to macromolecular complexes, such in the case of lipoprotein particles. So we came from this field to study pathology by using nuclear magnetic resonance. The high number of metabolites that this technology gives you, it allows you to apply a sophisticated computational approach based on machine learning methodologies to try to reveal what's happening in terms of clinical interest. So you can use the metabolome to obtain a risk stratification score by using regression modeling, so modeling a continuous variable from the whole set of variables that you obtain by using the technology. You can also apply this technology for clustering, so trying to identify which patients, which shared common features have patients with the same condition for pattern recognition. Or also you can also construct or build classification models for to construct a diagnostic test, so which biomarker is associated with a particular condition. And also as a predictive model, you can use the metabolome data to identify which phenotype will respond better to a therapeutical treatment and which patient will not. So this complete clinical ability of metabolome, it makes very, very interesting to be applied in clinical and actual clinical research. So I will present a bit what we have done before from the network, the research network that we have constructed through these late years with scientific societies, universities, and research institutes, mainly in Spain at the beginning. We are now expanding, but our first network has allowed us to explore NMR metabolomics in very, very different etiological processes and in different scientific questions, to answer different scientific questions. So we have applied metabolomic based on nuclear magnetic spectroscopy to evaluate different nutritional interventions, so with walnuts, pistachios, fish, even olive oil, lycopene, cacao fiber, postprandial response. So you can use the information from the NMR metabolomic dataset to evaluate what's happening after taking this nutritional intervention, also after taking pharmacological intervention with statins, with niacin, with fibrates, with inhibitors of PCSK9, with phytosterols, or even with omega-3s, and even in cancer immunotherapy, monoclonal antibodies. So metabolome can reveal what's happening after taking this placebo or pharmacological intervention, and monitoring this change in a quantitative way. As I said before, NMR metabolomics can be quantitative. It's indeed quantitative. We have also applied our technology, and in particular the lipoprotein profile approach, that it's one of the important modulators of the metabolism in exercise and disease interaction, trying to identify personalized programs that are better for a particular condition or not, and even in competition training, so which trainings are affecting in more efficient way to the energetic metabolism. But we have also used NMR metabolomics to evaluate internal factors, so genetic conditions, so hypercholesterolemia, hypertriglyceridemia with a genetic base, some dyslipidemias very specific that occurs with deficiency of a specific proteins as LCAT or acid lysosomal lipase, or also in glycerol kinase deficiency, that it occurs with a fake or false hypertriglyceridemia. We have also applied our technology to measure some other these beta lipoproteinemias, hyperkilomicronemias, even in animal models as a in knockout mouse. I'm trying to see what's happening in a atherosclerotic model for pharmacological studies. So, and at the end, I think that where we have more experience, it's per se to evaluate the disease. What's happening with the diabetes, with obesity, with chronic kidney disease, with liver disease, what's giving you the metabolome analysis in the polycystic syndrome, or even in infectious disease. In COVID, we have applied metabolomics in COVID, trying to see which are the different expression of the severity of the disease. In autoimmune disease, in for sure cardiovascular related disease, coronary artery disease, heart failure, premature cardiovascular disease, stroke, and in cancer, breast cancer, lung cancer, colon cancer, pancreas cancer, head and neck. And these molecules, in particular the lipoprotein profile, it's really reflecting that there's metabolic alterations that occurs years before the disease appears. So I would like to show you how the metabolome is altered during aging, and it's altered years before the disease appears. And for that, we thought that it would be interesting to prepare this specific talk about the early molecular signature of the disease. So trying to answer to this question, this particular question, is adult cardiometabolic health risk already determined during pregnancy? And for that, we had the perfect population study, because we had access to a very, very special cohort that included fetuses with fetal growth restriction. And it's particularly relevant because these individuals, this population in the adulthood, they will have more probability to develop metabolic syndrome, type 2 diabetes, obesity, hypertension, ischemic heart disease, polycystic ovary syndrome, renal disease, hepatic disease, so hypercholesterolemia. So it's exactly the population where this answer can be, this question can be answered. What is happening in these fetal growth restriction fetuses? Fetal growth restriction is defined as the failure to achieve the genetic growth potential. So it's a baby that it's small, and it's not small because of the parents are small. It's small because it has undernutrition, intrauterine undernutrition, and it affects around 6 to 10% of all pregnancies. So we studied a particular population in a collaboration framework with a fetal medicine research center that it's from Barcelona University. And it includes 3 groups of individuals, very newborn individuals, adequate for gestational age. So there was 28 newborns that were, in terms of birth weight, adequate. There were 25 individuals that were small, but not as extremely small as the third group that was a fetal growth restriction population below the percentile 3, and it includes 27 babies. We analyzed both the blood from the cord blood at the delivery, and we also analyzed the plasma from the mothers. So the material methods of this study were, we obtained the lipoprotein profile, the glycoprotein profile, and the low molecular weight metabolite and lipid species that can be obtained by using nuclear magnetic resonance from this cord blood plasma in the cases of the fetuses and from the mothers. Then we applied statistical analysis as a univariate and multivariate, trying to identify a specific molecular signature associated with fetal growth restriction or with the group. And then we try to see which kind of metabolites in general are affected altogether, trying to generate hypotheses that what's happening there. And I have adapted the principal result from this study in this table here. It's adapted from an already published article in Scientific Reports. That it's interesting because the most affected parameters that were associated with a fetal growth restriction compared to control individuals was the lipoprotein profile. In particular, I will explain you how to interpret this figure. It's representing the odds ratio, so the ratio between every one of these parameters according to the group. So in red, it represents which variables are increased in small fetuses compared to the control or adequate fetuses for gestational age in terms of birth weight. And in green, the opposite. So one of the parameters that it's green, it means that the control group presents higher levels. So the first surprising result was the pattern of the fetuses as opposite than the pattern of the mothers. So these parameters that are increased in fetuses are really decreased in moms. And that means that these fetuses that are very, very small, that present higher levels of very low density cholesterol, in remnant cholesterol, increased in remnant cholesterol, decrease in HDL cholesterol, known as the good cholesterol, but also an increase in triglycerides, in very low density lipoproteins, and increase in the number of LDL particles. So this pattern, that it's a clearly pro-atherogenic pattern beyond LDL cholesterol, it's exactly the opposite. So the mom, it's not able to adapt this catabolic state at the end of the pregnancy, and it's favoring an undernutrition of the fetuses, and this fetus should mobilize lipid metabolism, should increase lipid metabolism, trying to obtain energy from other sources. So this fetal growth restriction is clearly associated with an increased levels of triglyceride-related parameters in newborns. And in contrast, fetal growth restriction is associated with a decreased levels of triglyceride-related parameters in mothers. That suggests that a failure in the maternal metabolic adaptation to pregnancy that really makes a difference on the fetuses. These fetuses will, at the beginning, at delivery, they will have a pro-atherogenic pattern. So can we link these two patterns? Is adult cardiometabolic health risk already determined during pregnancy? The direct conclusions from this previous report that we have seen, and others that are concordant with this previous study that use nuclear magnetic resonance metabolomics technologies to understand the molecular signature associated with fetal growth restrictions, it's clear, the message is clear. This condition that dramatically increases cardiometabolic risk in adulthood occurs with a clear lipoprotein pro-atherogenic alterations at birth. And these pro-atherogenic alterations in lipoproteins, it's quite similar to the lipoprotein profile from adults with higher cardiovascular risk beyond LDL cholesterol levels. It's the profile, the specific signature of adults with atherogenic dyslipidemia. And what's atherogenic dyslipidemia? It's a lipid alteration that it's causing atherosclerosis, that it's increasing the risk of developing a cardiovascular disease in the future. As in the beginning we said that we were born trying to bring light to why is this population having cardiovascular accidents beyond LDL cholesterol. So we have to go a bit deeper. Several years of clinical research have demonstrated that the LDL cholesterol is not enough. Two individuals with exactly the same amount of LDL cholesterol can present totally different lipoprotein patterns, being this one here with the smaller particles that can cross this artery wall more easily, accelerate atherosclerosis, and increases the risk of developing cardiovascular disease. I will show you the concept of discordance between these two parameters. In this slide, in this figure from Journal of Clinical Lipidology, it includes more than 5,000 individuals that were followed 5 years or more, and it shows that it's really there's a heterogeneity between these two parameters. Can be a same individual with 100 milligrams per deciliter, that it's a correct level of LDL cholesterol, present more than the double of particles. In the case of here, these individuals with correct levels of LDL cholesterol, they have a higher LDL particle number. These individuals here are concordant in the diagonal. It's more or less the same percentile of LDL cholesterol than LDL particles. And in that case here, these individuals present a high level of LDL cholesterol, but the number of particles is not so much. It's reduced, in fact, because their particles are big. So if we stratify this population, these 5,000 people that was followed during 5 years or more, and we report the cumulative incidence of cardiovascular accidents, if we stratify these individuals according to the concordance between these two levels, discordance in the case of small particles or discordant in the case of large particles, even having in average less cholesterol in their LDL particles, they accumulate much more cardiovascular accidents because they have more particles. So the number of particles is a better predictor of a cumulative incidence of cardiovascular disease. That was very, very important in terms of cardiometabolic health, in particular in diabetic individuals or in obese individuals, because this pattern with smaller particles, it's much more frequent in diabetes and type 2 diabetes. And for that reason, since 2013, different clinical guidelines from the American Association of Clinical Endocrinologists or other clinical guidelines include the number of LDL particles into the risk stratification, and since 2016 as a therapeutical objective. So we were too simplistic thinking about LDL cholesterol can really discriminate or explain all these complex metabolism interaction that it modulates the atherosclerosis progression. We have been too simplistic. So in fact, there's more than LDL particles, because there's other particles that can be associated, that in fact are associated with increased risk of cardiovascular disease. Very low density lipoproteins content, both in cholesterol and triglycerides, that it's here in green and here in orange, increased the risk of a myocardial infarction or a stroke. You can see here as the line represent exactly the same risk for a group with elevated parameters or without elevated parameters. And in this case, you can see that people with elevated triglycerides has an increased risk of cardiovascular disease. People with increased levels of cholesterol in their IDL or intermediate density lipoproteins has an increased risk for myocardial infarction, ischemic stroke, and cardiovascular in general. LDL cholesterol increases the risk of cardiovascular disease, sure. But in the case of cholesterol transported in HDL, it's true, HDL cholesterol is associated with a diminished risk of future cardiovascular disease, but the composition of these HDL particles, if these HDL transport triglycerides, these triglycerides is also increase the risk of developing a future cardiovascular disease. So the lipoprotein profile beyond LDL cholesterol, it's quite relevant in terms of cardiometabolic health. In fact, nuclear magnetic resonance allows this direct from intact plasma or serum samples of the lipoprotein profile. In fact, the lipids inside the lipoprotein profile, the lipoproteins, resonate in the NMR spectrum in a univoc frequencies that allows them to be quantified. In the same way that the bells resound in a specific frequency and has a different sound, lipids inside the lipoproteins resonate in specific frequencies that make it possible to be quantified according to the size without having to ultracentrifuge the sample and separate the sample. So we developed this technology based on nuclear magnetic resonance that allowed us in a simple way to measure size, composition, and number of particles that are later associated with a cardiometabolic disease. It's a technology that it's fast, reproducible, it works from intact samples, it's high throughput, it's automated. So it has the requirements that clinical market needs. So we decide to constitute a company after patent the technology to industrially develop this technology to be applied in clinical environment. It was in 2013. We have obtained the approval from the Spanish Agency of Medication, the CE mark, and now we are trying to obtain the FDA certification. And in fact, we are already collaborating with different countries and different centers to try to implement and decentralize the technology for being used in clinical environments. We are already offering our technology through clinical laboratories, Spanish clinical laboratories as Echevarne, Synlab, Eurofins that are more European, and we are also included our technology in lipid units from different hospitals, in particular in Spanish hospitals. But now we are able to work by spectrometers that are outside of Spain. In South Africa, we have validated one spectrometer that can be used for measuring lipoproteins and other molecules from biological matrices. And in Chile, in Mexico, in Colombia, and even in Australia. So what are we offering exactly through these laboratories? We create a kind of a test that is a standard with a traditional parameters, so related with lipids, cholesterol, triglycerides, remnant cholesterol, etc., but also these advanced parameters that are associated with particles and size, and that are independently associated with future cardiovascular disease. In fact, that was too much information to be digested in 2 minutes in a clinical visit. So we decide to go a bit farther and create this silhouette, trying to illustrate what's happening to the lipoprotein profile beyond the traditional lipid factors. The silhouette contain the information of these parameters that are clearly associated with a cardiovascular disease, such the LDL cholesterol, but also the size of the particles, the cholesterol transported in HDL particles, the composition, etc. So if an individual has alterations associated with an increased risk in parameters, the silhouette is closing a smaller area, illustrating what's happening on their arteries. These three individuals present exactly the same amount of LDL cholesterol, but the risk of developing atherosclerosis and cardiovascular disease in the future is totally different. So I have selected a very, very small take-home message. NMR metabolomics is closing the gap between research and clinical applications. It's aligned with a precision medicine approach, and it exemplifies how far we can go applying preventive health strategies, even in newborn babies. We have clearly identified molecular proatherogenic patterns through advanced lipoprotein profiling and a special study group of newborns with a well-established adult cardiometabolic risk. So we have demonstrated that there's a molecular signature uh, before the disease, even when the child is born. But there's many questions still remain open. Are these molecular imprints irreversible? If not, can we modulate these molecular alterations? We still don't know, but I really believe that metabolomics will be the answer. And that was what I wanted to talk in this first deep tech talk. And thank you very much for your attention.